Wow, let's unpack this. For the last decade or so, the biggest revolution in cancer treatment has been immune checkpoint blockade, ICB therapy.
Right.
It's this idea that instead of, chemotherapy killing the cancer directly, we basically train your body's own T cells to do the job. The immune system's assassins. It's brilliant.
It is, but here's the frustrating reality.
Mm-hmm.
Something like 30 to 40% of patients, they never respond to ICB.
Wow.
And in really aggressive cancers, like bladder cancer or BCA, that number is even higher. Patients develop resistance. A lot of the time, it's because the tumor microenvironment is, it's cold.
Unresponsive.
Right. So you can give the T cells the green light with the therapy, but if the T cells can't actually get to the fight or get activated when they arrive, the whole thing fails.
You're missing the fundamental infrastructure.
Exactly.
For a successful immune attack, you need an organized command center, sort of like a temporary military base inside the tumor. We call these structures tertiary lymphoid structures, or TLSs. TLSs. Okay, help me visualize this. Is it literally like a mini lymph node that just pops up in the middle of a tumor?
Functionally, yes. That's a great way to think about it. A TLS is a transient, organized group of immune cells, T cells, B cells, dendritic cells that springs up where it's needed.
And its whole purpose is to activate those killer immune cells, the CD8 plus T cells.
At high efficiency, yes.
Yeah.
They are the engine room of the whole anti-tumor response.
So if we look at patient data, let's say from a big trial like the IMvigor210 data set, the patients with lots of healthy TLSs are the ones who respond well.
That is the rule. The data is very clear. TLS abundance is a decisive component for ICB response. But this leads to the big puzzle our sources were trying to solve.
Which is?
We know that the absence of TLSs means a resistant tumor. But what actively destroys them?
So we're not just looking for something that's missing, we're looking for a saboteur, an inside job.
Precisely. And this is where it gets interesting, because the culprit they found, it goes against a lot of previous thinking. B cells in tumors were often seen as, protective.
Right, the literature was kind of all over the place. Some B cells make helpful antibodies, others seem to dampen the response.
That ambiguity made them hard to target. But with these new tools like single-cell RNA sequencing and spatial transcriptomics, our sources could finally go on a hunt for the specific agent causing the collapse.
Okay, let's set the mission then. We are diving into the discovery of this specific B cell, the exact molecular mechanism it uses to sabotage the immune response, and crucially, how we might be able to stop it. So the first step for the researchers was to compare tumor tissues. They took samples from BCA patients who responded to ICB and from those who didn't. What did they see?
They confirmed what they suspected, a visible failure of that immune architecture, what they call a marked TLS decrement, in the non-responsive patients.
So the bases were crumbling.
Exactly. And since B cells are a cornerstone of any TLS, they focus their single-cell sequencing right there, on the B cells.
That's like knowing the base is failing and then deciding to read the personnel file of every single soldier inside to find the traitor. You need that incredibly high resolution to sort the good from the bad.
Absolutely. You can't just lump all B cells together, but cRNA-seq let them recluster all the B cells into 8 distinct groups based on their genetic signatures.
Yeah.
And they found a smoking gun.
An unusual group.
A very unusual group, which they called cluster B008. This group showed a significantly increased infiltration ratio, but only in the tumors that were TLS deficient.
They were showing up at the scene of the crime.
In high numbers. And this subset is defined by one marker that was way, way overexpressed.
Yeah.
Immunoglobulin lambda-like polypeptide 5, or IGLL5. So we now call these the IGLL5 plus B cells.
The IGLL5 plus B cells, the prime suspect. So where do they come from? Are they bad from the start?
Well, their origin is really revealing. Normal B cells that mature to fight infections usually go through what's called the germinal center, or GC, pathway. But trajectory analysis showed these IGLL5 plus cells came from a different route, the extrafollicular EF pathway. And even though they look like differentiated, mature B cells, they have this critical flaw,
which is
impaired humoral immunity function.
Wait, let me get this straight for everyone listening. These cells look mature, they're in a high-traffic immune area, but they're functionally broken.
That's a perfect way to put it.
Yeah.
They're like soldiers who've gone through training but can't actually load their weapon.
Wow.
They showed a reduced capacity to produce helpful antibodies in lab assays. So they're taking up valuable real estate in the immune structure, but they aren't contributing to the fight.
That explains why the old research was so confusing. A high count of B cells doesn't mean a high-quality response if these guys are in the mix.
Exactly. And this isn't just a bladder cancer problem. This is the really big finding.
It's pan-cancer.
It's pan-cancer. They looked at clear cell, renal cell carcinoma, non-small cell lung cancer, hepatocellular carcinoma, pancreatic cancer.
Yeah.
Across all of these really tough tumors, high IGL5 plus B cell infiltration meant fewer TLSs and much poorer clinical outcomes.
So they've potentially uncovered a fundamental mechanism of immune resistance that's shared across many different cancers.
That's the implication. We have our saboteur, the IGLL5 plus B cell. We know where it operates in these failing TLSs. Now for the big question, how? What molecular tool are they using to bring the whole structure down?
Okay, to understand the how, we need to zoom in on the heart of the TLS, the high endothelial venules, right? The HEVs.
That's right. If the TLS is the military base, the HEVs are the specialized fortified entry gates. It's how the lymphocytes, the T cells, get from the bloodstream into the tumor.
So if the gates stop working, no reinforcements can get in. The base starves and collapses.
Precisely. And the integrity of that gate is controlled by a crucial switch on its surface, the lymphotoxin beta receptor pathway, LTβR.
So, our saboteur B cell. How does it target this gate? This is where the spatial transcriptomics comes in, right? Mapping everything in 3D.
It was essential. The ST6 showed that these IGLL5 plus B cells aren't just wandering around. They showed a remarkable colocalization. I mean, they were physically stuck right onto the HEVs.
They're congregating right at the structural weak point. So how do they get there? Is there like a chemical GPS signal drawing them in?
Yes, there is. The immune system's GPS is its chemokine system. The HEVs, the gatekeepers, they secrete a signal called CCL19.
A beacon.
A beacon, exactly. And our culprit, the IGLL5 plus B cell, just so happens to overexpress the receptor for that specific signal, a receptor called CCRL2.
The CCL19 CCRL2 axis. So the gate is it's unknowingly calling its own destroyer right to its doorstep.
It's a perfect molecular hijacking. This chemokine axis guides the IGLL5 plus cells, lets them anchor onto the HEVs, and once they're anchored, they deploy the weapon.
Which is the IGLL5 protein itself.
Yes, the IGLL5 protein acts as the destructive ligand for that LTβR switch on the HEV surface.
And the researchers confirmed this was a direct interaction.
Oh, with incredible precision. They used multiple biochemical assays like pull-downs and mass spec that showed LTβR was the main thing IGLL5 was binding to. And then other assays confirmed a really stable, high-affinity bond between them. They found the key for the lock.
So what happens to the gate physically when this key turns in the lock?
Well, a healthy HEV should have a tight cuboidal morphology. Think of it like a strong, box-like structure.
Sturdy.
Very sturdy. But the HEVs that were next to these IGLL5 plus B cells showed a dilated lumen-like alteration. They became thin, weak, spindle-shaped. The gate isn't just closed, it's structurally collapsing.
Just completely compromising T-cell entry. The level of detail here is amazing. They even mapped the exact atomic handshake.
They did. Using AlphaFold 3 modeling, they pinpointed the exact amino acids. The LT core uses a sequence from Y88 to Q99 to lock into a sequence on IGLL5 from V148 to K158.
That's specific.
So specific that if you mutate just one of those sites, the binding affinity just plummets. They isolate the absolute tool of destruction.
Okay, so let's recap. The IGLL5 plus B cell uses a chemokine GPS to anchor to the HEV gatekeeper. It then uses its IGLL5 protein to bind and physically damage the LTβR receptor, causing the gate to collapse. Now, what is the signal that tells it to collapse?
This is where we get to the really clever part, the molecular flip. The normal job of that LTβR receptor is to activate something called the non-canonical NFB pathway.
Okay, and that is?
You can think of it as the HEV's maintenance crew. The receptor gets a signal, and it tells the maintenance crew inside the cell to keep the walls strong, keep them cuboidal, keep the gate open for business.
So if LTβR is the switch that calls the maintenance crew, what does IGLL5 do to that switch?
IGLF5 is an inhibitory LTβR ligand, and this is the core insight. Every other ligand we knew for this receptor, things like LTRA2 or LIT, they're all activating. They call the crew. IGLF5 does the opposite.
It's like a master key that when you put it in the lock, it turns the wrong way and jams the whole mechanism.
That's a perfect analogy. The binding of IGLL5 triggers a specific and dramatic conformational rotation in the receptor shape. Activating ligands shift it one way, but IGLL5 shifts the angle from about 120 degrees all the way to 180.
That physical twist is the sabotage.
It is. That change in angle flips the receptor's function from maintenance on to maintenance off.
And what's the immediate effect of that flip inside the cell?
It triggers a chain reaction. A regulatory protein called TLS3, which normally keeps the maintenance pathway in check, is supposed to be docked at the LT receptor. When IGLL5 binds and flips the switch, TLS3 dissociates. It just shuttles away into the cytoplasm.
And when TLS3 moves away,
it's no longer protecting a key maintenance protein called NIC, so NIC gets rapidly degraded. The whole maintenance sequence, that NIKP 100p52 NFB signaling pathway, is shut down cold.
the crew stops getting orders and the walls fall apart. You get that dilated, destructive shape.
The cascade is complete. The B cell gets recruited, it binds, it flips the switch, the maintenance crew is shut down, the HEV collapses, and the TLS starves.
And the final consequence for the anti-tumor fight is a disaster.
It becomes a highly immunosuppressive microenvironment. The activation of those anti-tumor effector CD8 plus T cells is suppressed. Instead, you're just left with a bunch of naive T cells that can't do anything. The immunotherapy is dead in the water.
So if the problem is this specific, the solution must be just as precise. Did they try to block the saboteur?
They did. They developed an anti-IGLL5 neutralizing antibody, let's call it IGLL5 100 other sites. And they tested it in very sophisticated humanized mouse models.
And what happened when they gave the IGLL5 antibody along with the standard ICB treatment?
The results were, frankly, stunning. The IGLL5 blockade did three things perfectly. It restored functional HEVs, it reversed the TLS destruction, and it reactivated those critical effector CD8 plus T cells.
Wow.
The combination treatment chemo anti-PD-1 plus the IGLL5 antibody was significantly more effective than the standard treatment alone.
It's like they cleared the road, rebuilt the base, and then the ICB treatment could finally get the army into the fight.
That's it, exactly. And this offers a huge therapeutic advantage. People have tried to fix HEVs before by directly activating the LTR with agonists, but that can cause a lot of vascular toxicity.
Right.
By just blocking the inhibitory signal, IGLL5, you're taking the brakes off. You let the system heal itself. It's a much safer, more elegant potential strategy.
So this deep dive, it really changes how we have to think about immune resistance.
It fundamentally reframes it. The B-cell population isn't one thing. It's a mix of good and bad actors. And it's not just about the presence of B cells, but the presence of this one specific subset, the IGLL5 plus subset, that acts as its own inhibitory checkpoint.
Sabotaging the whole immune response by jamming the HEV gateway.
Using that IGLL5 LTR axis.
And this finally resolves years of conflicting data about what B cells are doing in cancer. It proves that their specific type and their location are absolutely critical.
Right. While the preclinical data is incredibly exciting, the researchers are, of course, very clear that this needs to be validated in real-world human clinical trials.
Absolutely. But just think about that core mechanism we described. We have a protein, IGLL5, that flips a known immune receptor from an activator into a powerful inhibitor just by changing its physical shape, its angle of rotation. So this raises a really big question, something for you to think about. How many other immune evasion strategies might be relying on these subtle, sneaky, conformational changes to hijack our own signaling pathways? And what new key and lock therapeutics could we design to unlock them? What, to you, stands out as the biggest surprise in this whole complex story?